your future superstars out there. How are all my dear children doing? I hope all of you are doing fine. If at all there is someone who feels like it's too overwhelming, there are so many things to be covered, I have not yet covered it, don't be sad, you can still do it, okay? And I'm telling you, you can if you have the will to do it. So and understand this particular thing, that you have to be positive around things. Whenever things get bad, you have to think on the opposite direction. Think like, no, I can easily do it. This is very easy for me. I will do it and I'll show you how to do it. And then keep on going, my dear children. You will be definitely winning up that particular thing, whichever you are aiming for. So today in this particular session, I'm going to deal with the 1 marker question. And after finishing the 25 questions out there, only if you are finished, you have to put it down in the comment section that, ma'am, all the 25 questions are done. And now on the 26th question, you would have a bonus question that is left for you to do. So you have to do it only if you watch the whole video. You would have to put that comment also. Okay, so that's all. Are we ready? I hope we are ready. Let's get into it. So my dear children, according to 3 blueprints of the previous year, we see 1 MCQ question is definitely coming, and according to the 3rd blueprint, 3 MCQ questions have come from this particular chapter. So basically we have to be a bit prepared about the MCQ portions. But the good part of the story is that you're not going to get any MCQs out of the concepts that I'm going to tell you over here. So please make sure you're paying attention to all the small details and you are doing these questions with me. Okay, so the function of tapetum in Microsporangia is, okay, what is the function of tapetum? Where is tapetum? So my dear children, in the structure of an anther, you have different walls. Okay, you have different wall layers. Where is it? So the outermost is epidermis, then is endothelium, then is middle layer, and then you have tapetum. Inside the tapetum you have something which is known as sporogenous tissue, which is made up of so many microspore mother cell. Now what is your question? Your question is, what is the function of tapetum in the microsporangium? So in this whole structure, what is the function of this guy, that is tapetum? Now my dear children, this is very close to the microspore mother cells, which in future are going to form what? The pollen grains. So the function of tapetum would be what? Nourishing the pollen grains. So let's see what point matters the word. So it nourishes the developing pollen grains. Correct. It helps in dehiscence of anther to release the pollen. What do you mean by dehiscence of anther? So the anther has to break open and then release all the pollen grains which are going to be made up over here. So do you think this particular layer, that is tapetum, is going to help in the dehiscence of anther? Well, if you think so, yeah, that is wrong. Okay. So there are 3 separate layers, that is epidermis, endothelium and middle layer, which are the outer layers which take part in the dehiscence of anther. So now the question can be framed in another way. Which are the parts which helps in dehiscence of anther? So you know these are EEN, epidermis, endothelium as well as middle layer. Tapetum is just for nourishing. Now going ahead, it helps in protection, my dear children. Again, that is a function of the outer layers. Okay, it helps in division. No, it does not help in any sort of division. So basically your answer would be, it nourishes the developing pollen grains. Hence A is your answer. Okay, now going ahead with the next question. Select the correct sequence of anther wall layers. We just discussed it right now. What is the correct sequence of anther wall layers? First comes epidermis, the outside one. Then endodermis, inside, enter, right? So epidermis, then endodermis, then what? Middle layer, that comes in middle of what? Endothelium as well as tapetum. So again over here, if you see the picture, epidermis first, then endothelium, then middle layer, and then you have the tapetum. Epidermis, endothelium, middle layer, and then the tapetum. So your answer would be B. So one concept, multiple questions are there. Okay, now select the correct sequence of event in microsporogenesis, as in the formation of microspores, my dear children. How does it take place? What is the step-by-step process? So we saw in the sporogenous tissue, it is made up of lots of microspore mother cell. Now this microspore mother cell, what happens to it? It is going to undergo meiosis, and you are going to tell the step-by-step process. So we all know one thing, that there is porogenous tissue. There is sporogenous tissue which has what? The microspore mother cell. So here I don't see, so it's okay, but here I see, and here also I do not see. So probably these 2 options could be the correct one, but let's see, go ahead. So microspore mother cell is there. After that, my dear children, what do you have? 2 cells being arising out of it because of meiosis. You do not have any name for them. Now again, these 2 cells are going to divide into 2. What are these cells now? these cells are going to get clumped up together and they are going to be known as microspore tetrad. Tetrad means 4. So 1, 2, 3, 4. 4 of them are going to be together, my dear children. And individually, see, if you are speaking, they are what? Microspores. And these microspore, my dear children, later will be forming what? The pollen grains out there. So first, sporogenous tissue, where all this is happening. And in that tissue, one cell is there. Multiple cells are there, which are known as microspore mother cell. Microspore mother cell will divide and form microspore tetrads, and the microspore tetrad will form what? Microspores. So your answer would be what? This. If you see this, here microspore tetrad is given before microspores and what you call the microspore mother cell. So microspore mother cell is given later, what you call, what do you say that? Ranking, and we do not want that. This guy should be up over here. So your answer would be A. Okay. Now going ahead in the next question, the most resistant organic material present on exine of pollen grain is. What do you mean by exine? So exine is the outermost layer of the pollen grain. Pollen grain also has 3 layers. One is the exine, which is not continuous, it's a discontinuous layer. So they will be having pores like this. Okay, then is intine, which is continuous layer. Then the cell itself, my dear children, will be having its own plasma membrane. So plasma membrane, then your intine, then your exine. The most resistant organic material present on the exine of pollen grain is what? What is present in the exine, which is most resistant material? Pectin. Tapetum is a layer that we saw there. Sporopollen, cellulose. What is your answer? Your answer would be sporopollen. Remember, this is the most resistant organic material. If you put alkali, if you put acid, still it will be just like that. Which is why pollen grains act like fossils. They can stay up for a very long time, my dear children. So your answer is what? Sporopollen. It is found in the exine. Now in the inner layer, that is intine, it is made up of what? Cellulose as well as pectin. Cellulose as well as pectin makes up the intine. Exine is made up of this guy, that is sporopollen. And wherever you do not find this, it forms a pore that is known as germ pore. I spoke about all of this in the one-shot video. So if at all you have not watched it, watch it. Okay. Now going ahead with the next question. Which of the following has proved helpful in preserving pollen as fossils? Again, the same type of question but in a different way. So what feature is there in the pollen which allows it to stay like a fossil, which allows it to stay for a very long time? Again, the same thing only. It would be sporopollen. So your fifth answer would be D. Cool. Fine. Next question. Select the incorrect statement with respect to vegetative cell of the pollen grain. What is vegetative cell, my dear children? So if you see the structure of pollen grain, it looks like this. There is a big vegetative cell, that is this, my dear children, and inside the cytoplasm of the vegetative cell, there is a smaller cell out there, which is this. And this is known as what? Generative cell. Generative G, it is going to generate something. What is it going to generate? It is going to generate what? Gametes later on. Okay, it is going to form the gametes later on. So now you tell me what is wrong. You have to find the incorrect statement. Okay, with respect to vegetative cell of the pollen grain. So it is bigger than the generative cell. Is vegetative cell bigger than the generative cell? Yes, ma'am. Has abundant food reserve. Does it have? Yes. And it provides nourishment to the generative cell so that they can form gametes. Correct. Has large irregularly shaped nucleus. Now that is not clearly visible over here, but let me tell you, they actually have an irregularly shaped nucleus. So this is also correct. Now it floats in the cytoplasm of the generative cell. Does the vegetative cell float in the cytoplasm of the generative cell? No. The generative cell floats in the cytoplasm of the vegetative cell. So this one is the incorrect statement. Now if at all you are not reading properly, what happens? You are going to lose your marks as well. Okay? So read properly. And if at all you are not understanding the concepts very clearly, my dear children, I have made a detailed one-shot video. Go watch it. And if you are someone who have reached until the 7th question and have not subscribed to the channel yet, let me give you a small reminder. Please subscribe to the channel for getting all the updates. We are working really hard for you guys. If at all you are not making the most use of it, then for whom it is a waste? For you or me? Obviously you are missing out a great content. So definitely do not forget to hit on the subscribe button, and if you are liking, click on the like button as well. Now first thing first. So which one of the following statement is not true? Not true. Okay. Pollen grains of many species cause severe allergies. Do you think so? Yes, it does. Parthenium, which is known as carrot grass, that also causes severe allergy. Now stored pollen in liquid nitrogen can be used in crop breeding programs. So for example, I got a pollen of a plant which is soon gonna die. It is going to be extinct from the world. If I take the pollen grain and I store it somewhere, okay, store it somewhere, that is in the liquid nitrogen, at what? At minus 196 degrees Celsius. Please remember this degree, minus 196 degrees Celsius. Okay, if I store it up there, later on probably I can take the pollen grains and use it for crop breeding programs, that is for making of what? New crops out there. So this is also true. Now tapetum helps in dehiscence of anther. We just studied tapetum is there for what? Nourishment of the microspore mother cells out there. So over here this would be wrong. It's not there for dehiscence of anther. Now exine of pollen grains is made up of sporopollen, is it? Yes, it is actually made up of what? Sporopollen. So this is also correct. What is not correct? This particular statement, C1 is not correct. Okay? Tapetum helps in dehiscence of anther. Let me tell you feature about tapetum, that is it is usually binucleate. That means a cell can have two nucleus in nature. Okay, and it will have so much amount dense cytoplasm for what? For nourishing the microspore mother cells out there and the future pollen grains. Now the functional megaspore in an angiosperm develops into what? What is a functional megaspore? So just like how microspores are being formed which later form the pollen grains, my dear children, in the case of female plants, they have Megasporogenesis which is going to form the megaspore. So what exactly happens? What is a functional megaspore up over here? So the megaspore mother cell, just like how we had microspore mother cell, it undergoes meiosis. You have two cells. At the end you will have four cells, but then three of them are going to degenerate and only one is going to be surviving, and it forms what? It is known as the functional megaspore. And what does this functional megaspore do? Do you have any idea? It forms the female gametophyte, as in it is going to form something which is going to form gametes in the future, my dear children. So we call this as female gametophyte. So your answer for this question would be what? Embryo sac. What is a female gametophyte? It is the embryo sac. Okay, so this embryo sac later will be having what? The female gamete out there. So functional megaspore in an angiosperms develop into embryo sac, not endosperm, not embryo, not ovule. Yes. Okay. Next question. The following picture is an example for. The picture is given in the NCERT, but we usually tend not to see it. What is the picture depicting? So over here, these things that you see over here are carpels, my dear children. There are so many of them. That means the female reproductive system, which is also known as the pistil or the carpel. There are multiple carpels over here. So this is a multicarpellary condition for sure. Now the carpels, all of them, they are actually fused together. Okay, they are fused together. That means they all will be having a one ovary, my dear children. What do you call this condition as? Multicarpellary and apocarpus, gynoecium. Gynoecium is a female reproductive system. No, it's not apocarpus. Apocarpus means all of them, they are multicarpellary, carpellary, but they have different ovary. That is apocarpus condition. You see that in Michelia. There is a plant, Michelia. You also call it champak or chambagam. Okay, now over here it is multicarpellary and it is having what? Syncarpus condition. Syncarpus means all of them are fused. That is known as syncarpus condition. And what is this plant? This is papaware. No, Tupperware, just like that you have Papaware. Papaware is a drug, it's a plant which gives a drug, heroin. Okay, so Papaware somniferum, you're going to study it in human health and diseases. So your answer would be this, multicarpellary and syncarpus. It is not monocarpellary, there is no one carpel over here, there are multiple carpels. Not apocarpus, gynoecium, because all of them are fused, my dear children. Now going ahead, in angiosperms, microsporogenesis and megasporogenesis involves what? In angiosperms, microsporogenesis, megasporogenesis, we saw both of them. They involve what? Meiosis, we saw that, right? Meiosis was happening, one division, then next division. So it is common that they have meiosis. Occurs in the ovule? No. Megasporogenesis occurs in the ovule. Microsporogenesis occurs in what? The anther? No. So, no. Form gametes without the division. There is nothing in this world that can form gametes without divisions out there. So basically your answer would be A. Okay, children, understanding, getting, are you someone who is watching the one shot video and then have come to solve questions out of it? If at all you are someone like that, how do you feel getting the correct answer for all the questions? Definitely let me know. Okay, and if at all you find a doubt here and there, you can ask me that as well. Now, function of filiform apparatus is true. Where is filiform apparatus, ma'am? So over here in the picture of embryo sac, my dear children, you might have seen the synergid cells. If you zoom in a bit, you will be seeing a finger-like projection up over here. These are Filiform apparatus. Now, my dear children, this is the egg cell, that is a female gamete. So, the male gamete has to come through pollen tube. Now, who guides the pollen tube that, hey, you have to come over here, then only you can meet the egg? It is the Filiform apparatus that will be helping in guiding the pollen tube inside what? Inside the embryo sac, so that it can go and meet with the egg cell out there. So, what is the function of Filiform apparatus? Now, you tell me. To recognize the suitable pollen in the stigma? No, stigma is at a very distant position. Stimulate division of generative cells? No. Produce nectar? No. Guide the entry of what? The pollen tube. The pollen tube would be coming through it. And you know what? This is the best friendship story out there. Because the pollen tube is coming through a synergid, one synergid gets disintegrated, my dear children. One synergid gives away its life for its friend, that is the egg cell out there, the female gamete. But you don't need to give your life for any of the friend out there. You just need to make sure everyone is safe around you. Okay? Now, going ahead, the 12th question. From the statements given below, choose the option that is true for a typical gametophyte of a flowering plant. That means gametophyte over here, female gametophyte over here is what, my dear children? Embryo sac. So, what is exactly true for the embryo sac? That is the question. So, this is the embryo sac. This picture that you see over here is of the embryo sac. Now, you have to tell. I think I have put the same picture over here as well. So, let me see. What is already true? I have to see that, no? So, there are 3 antipodal cells at the chalazal end, okay? And there is 1, 2, 3. That is synergids as well as egg cell towards the micropylar end. You know, synergids and egg cell together, I call them as egg apparatus. So, all of them are towards the micropylar end. I told in my video, one short video, the micropylar end is very important. It is important because of this reason. And then, my dear children, there is a big cell out there which is known as central cell, and the central cell over here, my dear children, will have 2 polar nuclei. There is 2 polar nuclei. So, if I talk about the nucleus 1, 2, 3, the red one is a nucleus. So, 1, 2, 3, 4, 5, 6, 7, 8. 8 nuclei are there. But what about the cells? So, 1, 2, 3, 3 antipodals, 1, 2, 3 synergids and X 6 plus 3 plus 3, 6. And then this whole thing is what? 7th cell, that is the central cell. So, 7th cell is there, 8 nucleus is there. So, you tell me now. It is 8 nucleate and 7 celled at maturity? Correct. Sometimes this can be given ulta. 8 celled and 7 nucleate. Don't get confused out there. Read the question carefully. Now, it is free nuclear during the development. What do you mean by that? That is basically, I told you, one functional megaspore is the one that is going to form what? The whole embryo sac. And it is not a very direct process. Initially, the nucleus of the embryo sac, nucleus of the functional megaspore, it undergoes division. First mitotic division, mitotic, not meiotic, okay? First mitotic division, then again the nucleus will divide, second mitotic division, then again the nucleus will divide. At the end you will have 8 nucleus, and this is known as the third mitotic division. So, 3 mitosis happens, and over here the nucleus are dividing. Just after the nuclear division, you don't have a cell wall formation. It waits for some time for the formation of cell wall. And once the cell wall are being formed, my dear children, the cells assemble towards either side. So, antipodal cells, then come synergids, then come egg cell, and then you have the polar nuclei, which is a part of what? The central cell out there. So, here the thing that is given is what? Just a second. It is free nuclear during the development. Is it? Yes, free nuclear means it is not bounded by cell wall immediately. So, that is also correct. It is situated inside the integument but outside the nucellus. It is situated inside the integument. So, if you have seen the structure of the ovary, my dear children, you might have seen that there is ovule over here, and the ovule will have its own integument. Right, these are the integuments, and inside the ovule there are certain cells which you call as nucellar cells, and inside that all of this is happening. So, it is situated inside the integument, yes, but outside the nucellus, no. In the innermost area it is happening. So, it is not outside the nucellus, it is inside the nucellus as well. So, this is wrong. It has egg apparatus situated at the chalazal end. Egg apparatus, is it at the micropylar end or the chalazal end? It is towards the micropylar end. So, again this is also wrong. So, if at all you do not know all these details, will you be able to answer it? No. So, your answer would be what? A and B. You have to tell what is what true. So, A and B are what true. So, your answer is C option. Just a second, now all the animations will be coming. Yeah, that's it. Okay, now going ahead, identify the correct sequence of arrangement of cells in the mature embryo sac from the micropylar end. Again, you do not know what's there in the micropylar end, then will you know the answer? No. So, first of all, you have the synergids, then the egg, then the central cell, and then the antipodals. So synergids, then the egg, then the central cell, then the antipodal cells. Your answer would be A. If chalazo line is being asked, then antipodals, central cell, then egg cell, then synergids. Ulta of it. Okay? Cool? Next question. Which of the following is not a correct statement? Not a correct statement. You have to find the wrong statement. Placenta is located outside the ovarian cavity. So my dear children, this thing that you see is the ovarian cavity. This is the ovary, and the cavity which you see over here is the ovarian cavity. And this is the ovule with all its integument. And over here you will be having the placenta within the ovarian cavity itself, and it is connected to the ovule with the help of something that is known as funicle. So those of you who have attended the one-shot lecture know the answer that placenta cannot be located outside the ovarian cavity. It is within the ovarian cavity. This statement itself is not the correct one. Central cell develops into primary endosperm cell after fertilization. Does it? The central cell which is there, okay, does it form primary endosperm cell after fertilization? Yes, it does. And this is known as triple fusion. What is happening? There are two male gametes being formed. One male gamete fuses with the egg cell to form the zygote. The second male gamete comes and fuses with the two polar nuclei. Okay, now you have two polar nuclei and the male gamete's nucleus. So three nuclei are there. N haploid, all of them are haploid. So triploid nucleus is being formed over here, and the cell itself where all this is happening, it transforms into what? Primary endosperm cell. So this is also correct. Okay, now orchids contain more than one ovule in an ovary. Correct. It has multiple ovules. It's not like mango which just have one ovule. Always remember, mango is something that has one seed, no? So if it has one seed, that means it is one ovule also. Okay. Not always, but generally. Now, egg apparatus consists of one egg cell and two synergid cell. Yes, ma'am, I know that. So your answer would be A. A is the odd one out. It is the one that is not correct. Now going ahead, even in the absence of pollinating agents, seed setting is assured in. You do not need any pollinating agents, but seed will definitely be formed. How, ma'am? Miracle. Well, it can happen if the flower is closed and the flower has both the female as well as the male part. Both female as well as the male part is there, and if the flower is closed, my dear children, do you think you need pollinating agents? No. Over here the pollen grains will land up over here and do the whole thing, right? They will do the whole thing. So even in the absence of pollinating agents, seed setting is assured in which plant? Do you know which plant have this closed type of flowers? Cucurbita, papaya, commelina, maize. Your answer is what? Commelina. So commelina, viola, oxalis, they all have clistogamous flowers. They all have clistogamous. CL stands for closed. They all have closed flowers, and there is one more type of flower that is known as chasmogamous flowers. That are the open ones. Okay, that are the open ones. So the open ones are more towards the upper side, and the clistogamous, the closed ones, are more beneath the soil or just above the soil. Okay, towards the rhizome or root or just above the soil. So your answer is what? C, my dear children. Okay. Next. Transfer of pollen grains from the anther to the stigma of another flower of the same plant is known as. Transfer of pollen grains from the anther to the stigma of another flower but of the same plant is known as gytonogamy. Okay, gytonogamy. So if it is happening within the same flowers, the same flower has both the female as well as male part, this is autogamy. If it is happening between two flowers, my dear children, this is known as gytonogamy. If it is of the same plant. If it is of a different plant, then it is known as xenogamy. Cross X, xenogamy. So over here it is gytonogamy. Your answer would be D. Cool. Fine. Next, which pollination brings genetically different types of pollen grains to the stigma? Genetically different type. Does gytonogamy bring? No, ma'am, it's between in the same plant. No. So obviously no. But can xenogamy bring genetically different type of pollen grains? Yes, because it is happening between two different plants. So your answer would be xenogamy over here. Okay? Not gytonogamy, not autogamy, not clistogamy. Clistogamy does not play any role over here. So your answer is B. Next, dioecious plants are, what do you mean by dioecious plants? Dioecious plants are the one which have separate sexes. For example, one plant will be all female, another plant will be all male. These are dioecious plants. So over here, my dear children, what type of pollination do you think would happen? Can autogamy happen? Autogamy means from the male to the female part of the same flower. Gytonogamy, from the male to the female part of two different flowers of same plant, it does not have the male part itself, then how can gynoegamy happen? So we have only one thing can happen, that is true cross-pollination. And what is true cross-pollination? Gynoegamy is also cross-pollination, but it is not true. The true one is only one thing, and that is xenogamous condition. So your answer, dioecious plants, like what? Example you need to remember, dioecious like papaya. Papaya is a dioecious plant. It would be having only xenogamous, and this is one of the best way to produce genetically different type of plants. Now, which is not a characteristic feature of the wind pollinated flowers? Feathery stigma. Yes, it should be feathery, otherwise how will it catch the pollen grains? Presence of a single ovule in the ovary. Yes, the flowers which are there, they will just have a single ovule, no multiple ovules. Why, my dear children? Why to waste too much ovules? Because wind pollination is not a guaranteed thing. That is the reason. Now, pollen grains are non-sticky. Obviously, if at all they are sticky, they will go and stick somewhere rather than moving around in the wind. Now, flowers are very colorful. Is it so? What is not the characteristics of wind pollinated flowers? For the wind does not know colorful or not. So over here, flowers need not be colorful, my dear children. So this is something that is odd one out, D. Cool. Next question. Wind pollination is common in. Wind pollination is common in. It's a very important exam. Legumes, lilies, grasses, orchids. Your answer is grasses. Again in the one shot I clearly told you. One more example is there. Anyone who has listened to the one shot video, can you tell me the one more example of wind pollinated plants? Apart from grasses, there is one more. Come on, guess, keep on guessing that and put it down in the comment section. I would love to know that. And 21st, in a fertilized embryo sac, or if at all you have not watched the one shot video, can you just open your NCERT and check it out and let me know? Can you do that? You should be doing that. You should know what are you missing, and that's why I'm telling you watch the one shot video. Now, in a fertilized embryo sac, the haploid, diploid and triploid structure are in the fertilized embryo sac. So my dear children, the egg cell is being fertilized. The egg cell having n number of chromosome is being fertilized by the male gamete also having n number of chromosome, and now n plus n you get 2n, and 2n is what? What are you forming when male and female gamete comes together? You form the zygote. So zygote is diploid in nature. This is what? Diploid. Now another male gamete is also there. So this one is having n number of chromosome, this polar nuclei is having n number of chromosome. The male gamete which is coming here, its nuclei will also have n number of chromosome. So n plus n plus n, it forms what? A triploid nucleus over here. A triploid nucleus, or can I say a triploid cell? A cell that has a triploid nucleus, right? What do you call this cell as? You call this as primary endosperm cell, PEC. Primary endosperm cell, PEC. And what do you call this nucleus? Primary endosperm nucleus. You call this nucleus as primary endosperm nucleus. So what is triploid over here? The primary endosperm nucleus or the primary endosperm cell. Now what about haploid structure? Everything else is going to be haploid. The synergids are going to be haploid. The antipodal cells are also going to be haploid. They didn't have anyone coming and fertilizing with them. So you have to tell what is haploid, what is diploid, what is triploid structure. So synergid is haploid, zygote is diploid, and primary endosperm nucleus is what? Triploid in nature. Here synergid antipodal is also haploid, so this is wrong. Synergid polar nuclei, if we talk about one polar nuclei, it is n in nature, my dear children. If we talk about both of them, it is 2n, but not, okay. So anyway, you're talking about one polar nuclei, so obviously it will be n. It is haploid, so it is wrong. Antipodal synergids, again synergids is what? Haploid, so not approved. So only one thing is there, and that is your answer A. Okay. Next, endosperm persists in the mature seed in. Endosperm, there are some plants in which the endosperm is still there, which helps in nourishment of zygote to the formation of embryo. Okay, and after forming the embryo also it still persists somehow. What are those plants? These plants are known as monocots. These plants are also known as the plants which have albuminous seeds. These are also known as the plants which have albuminous seeds. Example, groundnut, is it? No, it's a dicot. Castor, is it? Yes, it is a monocot. Pea, is it? No, it is a dicot. Beans, is it? Is a dicot. My dear children, whatever things you can break into two, they are dicots because they have two cotyledons. Okay, so groundnut, pea, beans are example of dicots. Remember that. Castor is monocot, and castor is something that is known as albuminous seed because it does have endosperm still persisting. Now, ma'am, what are the others? They do not have endosperm in them. What do you call them? What type of seeds do they produce? They produce non-albuminous seeds. So if at all again a question comes, what among them produce non-albuminous seeds? You should spot the dicots. You should spot the dicots. Do you have another example for monocots apart from castor? Wheat, barley, rice, okay, coconut, all of them are monocots. Now next question, in a monocot embryo, shoot apex and leaf primordia are enclosed in a hollow foliar structure known as, so shoot apex, which is going to be forming the shoot, my dear children, the shoot region, and the leaf primordia, all of it is covered by something which you call as what? Coleoptile. You call this structure as coleoptile. Coleorhiza is towards the root. So R is there, so it is towards the root. And over here P is there, so it is towards the plumule, which is also known as the shoot apex. You can remember like that. So your answer for this question would be coleoptile. Amma. Fine. Chalo. Next question. Perisperm differs from endosperm in. Perisperm, how do you think it is different from endosperm? Being a diploid tissue, what is perisperm first of all? What is perisperm, my dear children? So in the ovary, I told you there is ovule. In the ovule, there are certain cells which are known as nuclear cells. Just like how you have sporogenous tissue, right? But in sporogenous tissue, all of them are microspore mother cell. Over here, only one megaspore mother cell is there. Rest of the cells are still nuclear cells only. And usually they go off. Okay, they go off, and whatever has to happen will happen. That means embryo sac is going to be formed, everything happens. But in some cases, the nuclear cells still persist there. And this persistent nuclear cell which is there, it is known as the perisperm. It is known as the perisperm. And what is the ploidy of perisperm? It is just like another normal cell of the plant, my dear children. So it will be diploid in nature. So perisperm differs from endosperm. Endosperm we saw it is triploid. Triploid. Here it is diploid in nature. So perisperm differs from endosperm in being what? A diploid tissue. Being a haploid tissue, no. Having no reserve food, no. It has reserve food, that is how it is nourishing. Its formation by fusion of a secondary nucleus with several sperms, no. Your answer would be A. Okay? Cool? Chalo. Next. Which part of the fruit labelled in the given figure makes it a false fruit? So this thing is given. What is this first of all? Let me tell you what is exactly this thing. This is the last question. After that, there is a bonus question for you guys to solve. So this thing that you see is an apple, okay? And over here, these are the seeds, that is the ovule. Just around the seeds, you have the ovary. And outside the ovary, you have something else also which is taking part in the formation of a fruit, which is why this is known as a false fruit. What do you mean by false fruit? When it's not just the ovary, but other parts also contributing for the formation of fruits. So what is this other part that is making this as a false fruit, my dear children? It is the C part. And what is C? What is C over here? It is thalamus. Okay, so thalamus means this structure onto which everything is resting in the plant, no, in the flower. So this is known as thalamus. So thalamus is also contributing in the formation of fruit over here. So it is not the seed which makes the false fruit. Fruits, all the fruits have seeds, my dear children. It's not the mesocarp. Mesocarp means what? The walls of the ovary. In the walls of the ovary, the outermost layer is known as epicarp. Then you have mesocarp, and then the one that covers the seed is known as endocarp. You can clearly see in mango. In mango, the thing that we peel off, it is the epicarp. Then what we eat is mesocarp, and the one that covers the seed, the hard shell sort of thing, is the endocarp. The seed has its own seed coat, but I'm just telling you. So over here, everything is there. Endocarp, epicarp, mesocarp, everything was over here itself. And outside that, you have the thalamus, which makes it the false fruit. I'll show you the actual picture. Oh yeah, where is it? I didn't put the picture. It's okay. It's okay, it's there in your NCERT. I have mentioned in the one-shot video as well. But yes, this is the thalamus. I've given you the answer. Yeah, and now one question which is there for you guys to solve, and that is this. Statement 1. Formation of a fruit without fertilization is known as apomixis. Is it true? Formation of fruit without fertilization is called apomixis. I'll give you a clue. No, it's not true. I'll tell the answer itself here for the first statement, okay? First statement is false. Why? The formation of seed without fertilization is known as apomixis. Formation of fruit without fertilization is known as parthenogenesis. There the ovary is turning into a fruit, but the ovule is not turning into a seed. So formation of fruit without fertilization is known as parthenogenesis. It happens in banana. That is why banana does not have a seed. Okay, so you can make seedless type of plants with the help of that. Now statement 2, in some species of Asteraceae and Grasses, seeds are found without fertilization. That's your turn to answer. So tell me the whole answer for this question in the comment section, and when I see it, I'm going to tell you I'm really happy if I see it. Okay, I will be happy, sorry. I will be happy if at all I see your answers coming out there. And my dear children, I think whatever we are doing as of now, it is useful for you, isn't it? Okay, if it is useful, definitely let us know. Share it with your friends if at all you have not shared it yet. And my dear children, if at any point you feel that you are not able to do, there is too many backlogs, cover what is there in front of you, cover the high weightage chapters. It's very important. That's all I want to say. And I am completing the high weightage chapters one by one. So if at all you are worried about what is going to come in front, I'd say not to be worried about that much. Be worried about what is being completed and go revise it whenever you can. Okay, so that's all. Wishing you all the best. Lots of love. Bye-bye. Take care of yourselves.